Okay, so in the last two classes, we have discussed about floral biology. I've given a complete overview of floral biology in the previous two classes. So this is the third class of seed science and technology, and in today's class, we are going to start our discussion about seed physiology. Okay, so it might take one or two classes to finish this seed physiology. I'll just give you the outline of what are the content that we are going to cover. Okay. So once this is done, we can just shift into seed production, then seed testing, seed production, and post harvest management or post harvest operations, then seed testing. Yeah, then we are done. Okay. So seed physiology. Just to give me a second, I'll just add some blank pages. Outline the events, or I outline the content that we are going to cover, or I'm going to cover in this seed physiology class. So starting with, we are going to discuss about germination process of germination, and secondly we are going to discuss about plant hormones or growth hormones, and thirdly we will be discussing about dormancy, and before dormancy we'll just talk about reserve seed mobilization, or generally we will talk of seed reserves, seed storage reserve materials. So these are the contents from your ICAR JRF syllabus, that is under seed physiology. So we will stick our discussion for mainly these four topics. Okay. So to start with, can anyone tell me what you understand by this seed physiology, or what are the aspects that are covered under this seed physiology? As you can already see that I have written the outline here. So what do you understand by seed physiology then? So seed physiology is a branch where it deals with the understanding the germination and the seedling emergence. See, whatever that we study under seed physiology, it is more or less or broadly related to either germination, not either, germination and seedling emergence. So we will be studying all the aspects, all the changes, and all the factors that are influencing the germination and the seedling emergence. So all these mechanisms and concepts will come under seed physiology. So it basically deals with the understanding the germination and seedling emergence, and also the relationship that are influenced by the changes in the environmental factors. So what are the environmental factors that can influence this germination and the seedling growth? Mainly it is temperature, moisture, oxygen, and light. And also we have some germination inhibiting substance or the chemicals, and also the phytohormones. So all these things are covered under seed physiology. Hope this is clear. So till previous class or till yesterday's class, we have discussed few things about the germination and all. So few content might overlap between the floral biology and seed physiology. Okay, so few of the things I have already covered in the floral biology. I'll just take them forward now. Okay, so it's about germination. So we have discussed until the formation of the mature seed, where it will undergo the different phases or different stages, where we will get a matured seed or fertilized ovule, which is ready for the regeneration of the plant, from which the new plant can regenerate. So this particular seed is actually capable of germination, or even when this particular seed is in the plant itself, so this will be under hydrated environment, right? So there will be in the hydrated environment even when the harvesting is not taken place. And the second thing is about germination. We know that for germination to take place, it requires imbibition or hydration. So once there is the imbibition, the seedling will emerge or the radical protrusion takes place. So my question is, even if the seeds are under the hydrated environment within the plant, why don't they germinate usually? Yes, can anyone answer? So my question is, seed development, whatever the development process of the seed, that will take place on the mother plant, right? So all this development is under the hydrated environment. So though it is in the hydrated environment, it won't germinate within the plant. It is only after the harvest and after sowing it in the field, it will germinate or the radical emergence takes place. So what is the main reason for it? What are the factors that is inhibiting the germination on the mother plant? Yes, dormancy. Okay, dormancy, ABA. Nice, good. Anything else? Any specific reason why the seed will not germinate within the mother plant? You all are speaking about the result, like how the germination is not occurring, how it is inhibited. So it is basically inhibited by the mechanism of dormancy. That is true. But why this inhibition is required? Why on the mother plant germination should not occur? Yes, a medium is needed. Okay. Yeah, see, the simple medium for the germination of any seed is water. If they imbibe the water, they will have some seed, seed storage reserves in them where it will mobilize and the enzyme activity will begin, and there will be a radical protrusion. So this is a simple thing. It all depends on the imbibition. So I told you, like even in the hydrated environment, seed should not germinate. It won't germinate. Why? What is the reason? You already told me it is because of dormancy that it won't germinate. So why it should not germinate, or why the plant has adopted a mechanism of dormancy? Why do you think this mechanism has been adopted by the plants? Yes, anyone? Okay. So, for my question, why there is no germination even in case of hydrated environment? So this is basically for three main reasons. One is, this is the main and first and foremost reason why germination doesn't occur. So how this germination is independent? This is inhibited by dormancy, the mechanism of dormancy that we have understood. But why? It is because of lack of nutrients that are accumulated in the seeds. So the nutrients that are accumulated, or the process of accumulation of the nutrients, will be taking place during the seed development. So since there will be no full accumulation of these nutrients, if they germinate, they will be non-viable. Even if the radical protrusion takes place, they cannot give rise to complete plant. So they simply means that they are non-viable in this case. So this is the first and foremost reason why germination doesn't occur in the mother plant itself, because of lack of nutrients or storage reserves. Secondly, it is because of constraints imposed by surrounding tissues. Can anyone just tell me the examples of the constraints that are imposed by surrounding tissues? Name of the parts which will impose the restrictions on germination. Constraints by the surrounding tissues of a seed. What are the surrounding tissues of a seed that you know? Yes, exactly. It's seed coat, integuments, testa. All these are surrounding tissues of a seed. So if there is a hard seed coat, so it won't germinate. We all know that, right? So it is called as seed coat dormancy, or the dormancy induced by seed coat. So this is the second reason why germination cannot occur on the mother plant, because there will be constraints that will be imposed by the surrounding tissues. Thirdly, it is because of presence of ABA. So for my third point, that presence of ABA, does this mean that the maternal ABA is contributing to the seed, or this is transferred into the seed and this is acting on a seed, or it is the embryonal ABA that is already been synthesized and this is acting on it and it is inhibiting the germination? Which one is true? Is it is a maternal ABA or the embryonal ABA? What do you think? Like embryo can synthesizes ABA while during the development or during the seed development, can you imagine that embryo itself is synthesizing a plant hormone which is nothing but ABA, or it is the maternal ABA which is giving the support to the embryo where it is inhibiting the germination. So it is basically the maternal embryo which is inhibiting the germination. So what is the germination called as when it occurs on a mother plant? There is a specific term for it. If there is a germination of the seeds on a mother plant, you all know it, but you are not able to recall it, I think. Yes, it's vivipary. Can you just give me the broader term? It's precocious germination. So it is because of precocious germination, or the germination if it takes place within the mother plant, so that is called as precocious germination. So even in this precocious germination, we have two different mechanisms. So one is called as vivipary. So this type of clarity, people might not be having it. So since we have studied this in our MSc, I have bit clarity about what is the difference between the vivipary and what is, like, if someone comes to me and asks me what is precocious germination and what is vivipary. So this is nothing but the same. I won't be able to tell them that okay, this comes under this precocious germinations in that particular stage. But since I'm a PhD student, I know it like this is a classification under the precocious germination. So one is vivipary. What can be the second one? Can anyone guess? So this is pre-harvest sprouting. Have you heard about the term pre-harvest sprouting? Yes. Okay. So in this particular slide, what I have discussed with you that how the germination is inhibited, and this is mainly because of three main reasons. And even though there are some mechanisms, there are three main mechanisms of how germination is inhibited. In some of the plants, we see the precocious germination, right? So this precocious germination has again two types. One is vivipary, and second one is pre-harvest sprouting. So this is the assignment for you. In the next class, you should tell me the difference between vivipary and pre-harvest sprouting. So if you already know it, you can just either chat or text in the chat box, or you can unmute yourself and answer. So this is about vivipary and pre-harvest sprouting. So make sure to remember this and search for it. If you get the answer, come back to me, because if you just search for this answer, you will not be forgetting it further. Okay. So this is about germination or germinability during the seed development. And there is certain condition where you will see in the fleshy fruits. When I talk of fleshy fruits, for example, tomato, so seeds are inside the flesh, right? So in this type of fleshy fruits, you already know that the seeds are already in the hydrated environment, or seed development itself is taking place in the hydrated environment. Yet you don't see a germination inside the fruit. Inside the tomato fruit, you don't see the germination. So what is the reason for it? Or the germination is hampered? Again, it is something related to ABA. One reason is ABA. It is because of the ABA, which is from the maternal ABA, that this germination is inhibited. There is also the other reasons in case of fleshy fruits. What is this? So what will be the osmotic potential of the surrounding of the seeds, whether it will be high or low? In the tomato fruit, if you just imagine the tomato fruit, the seeds that will be present inside the fruit, the surrounding tissues will have the higher osmotic potential or lower osmotic potential. So how do you define this osmotic potential? It is nothing but the solute concentration. So since it is made up of flesh and it has different nutrients, the solute concentration will be very high. So the outside osmotic potential around the seeds will be high in case of tomato fruit. So if this is the case, whether the water will move outside the, like move from inside to outside or from outside the seeds to inside the seed, one or two. In case of higher, like if the surrounding tissues is having higher osmotic potential than the seeds, whether the water will move from inside the seed to outside or outside to inside. Yes, it is from lower osmotic potential to higher osmotic potential. So we have already discussed that if the germination has to occur, seed has to imbibe the water. Only after the imbibition of water, all the enzyme activity can begin, and there will be the digestion of the storage reserves, and this is how it will get the energy, and it will be able to protrude the radicle. So this is again inhibited because of high osmotic potential. So this will be the second reason, high osmotic potential of the surrounding tissues. So this will inhibit the germination of the seeds. I hope this is clear. So these are some basic concepts. So if you can expect some questions based on all these concepts, might be the direct questions or indirect, but they might ask you few things about it. Okay, make sure to remember. So this is all about germinability of the seed development or during the seed development. Now we can start our discussion about plant hormones. Can you name the important plant growth hormones or growth regulators in case of plants? Or like before just naming the plant growth hormones, so what are these plant growth hormones? What is the function of these plant growth hormones or plant hormones? What is the major function of these growth regulators or plant hormones? Yes, it's auxin, cytokinin, ABA, gibberellins, ethylene. Very good. So what is the main function of these plant hormones? So these are nothing but the signal molecules. They will transmit the signal from one part of the plant to other part, and they are produced within the plant. These are, that is why they are called as plant hormones. And thirdly, they are very active at very low concentration. And fourth, they have the important function to regulate the cellular process or regulate cellular and developmental process. So this is the main function of the plant hormones. So now we'll talk of important growth hormones. So mainly there are 6 hormones. One is ABA, auxin, gibberellin, cytokinin, ethylene, and bassinosteroids. And there are also the other hormones which are jasmonates, carikins, and strigolactins. So these carikins are mainly found in smoke. Where in case of forest fire, the germination of the plants after the forest fire is mainly because of the hormone carikins. Okay, so you need not to worry about all other hormones. Even though I have just mentioned few information or written some information regarding these hormones, you can just go through it once. So we'll stick our discussion to mainly these 6 hormones, out of which we will discuss ABA and auxin in detail. So these are the 2 important hormones which have the role to play in case of growth and development. So, but when we consider germination, it is ABA and the abscisic acid, or the concentration of the ABA and abscisic acid, which will decide whether the seed will germinate or not. So these are nothing. These are simply in opposite in the concentration, opposite in their function. So if ABA content is high, then the GA content is high, then ABA content will be low. If ABA content will be high, then GA content will be low. So in the first case, if GA content is high and ABA content is low, germination occurs. And in the second case, if the ABA content is high, the germination is hampered or inhibited. So I've made a list of the functions or the roles of different hormones, starting with abscisic acid, auxins, gibberellins, cytokinin, ethylene, and bassinosteroid. Just go through these roles once. We'll get to understand them better. Okay. So what is left? So we'll just talk of auxin synthesis now. How this auxin is synthesized in the plants? What is the precursor of this auxin? In the higher plants or in the plant itself, which is a precursor of the auxin? Or before the precursor, which is a main auxin in case of the plants that you see? Have you heard about the name IAA? Yes, very good. It's tryptophan. The precursor of the auxin is tryptophan. So it is, yeah, indole acetic acid, which is the main auxin in case of higher plants. Yes, very good. No, it's not methionine. It's tryptophan in case of higher plants. Okay, this is clear. So the precursor is tryptophan. That means from the tryptophan, the auxin or IA is synthesized. So this synthesis is a two-step process where tryptophan will be converted into indole 3-pyruvate. Someone has raised your hands. Okay. So indole 3-pyruvate. In the second step, this indole 3-pyruvate is converted into IAA. So there are two main enzymes here. One is transaminases in the first step, and secondly it is, I don't know whether you have heard about this enzyme or not. It's UCAR, YUC mono, like YUC flavin monooxygenase. This is the enzyme which will convert this indole 3-pyruvate into IAA by the process called, or by the reaction called decarboxylation. Here from the tryptophan, the amino group is removed. So the removal of the amino group from the tryptophan, if you just see the structure of the tryptophan, it will be converted into indole 3-pyruvate. This is just a two-step process of how the auxin is synthesized in case of higher plants. So talking about the function. So if I just talk about the body level, the enzyme, endosperm, and the seed coat. So this is 3N and this is 2N, and again this is 3N. Right? So this is enzyme, endosperm, and the seed coat. Sorry, this is 2N. So this seed coat is the maternal tissue. So what I mean to say is these are genetically dissimilar, right? They are not genetically same. But during the development of the embryo and the endosperm, or during the development of the seed, you see that there is some coordination between all the three different parts. So auxin is mainly responsible for this coordinated growth. Even though they are genetically dissimilar, it is because of the auxin there will be a coordinated growth between these three different parts, or these three different organs. This is the main function of this auxin. About the synthesis, can you tell me whether the auxin synthesis will takes place before germination, or sorry, before fertilization, or after fertilization? So auxin synthesis will only takes place after fertilization, not before fertilization. It is after fertilization. Okay, because only after fertilization it should germinate. If this auxin is synthesized before fertilization, so there are the chances that the female gametophyte or embryo itself or embryo sac itself can germinate. So that is why the auxins are always synthesized after fertilization. So how this synthesis is regulated? This is by the certain class of genes which will inhibit the synthesis of auxin when the fertilization is not even taken place. After the fertilization event, signals will be passed where this gene synthesis will be blocked, and another set of genes will be synthesized, which will help in the synthesis of this auxin. So this is about the auxin function. So if I want to specifically tell you about the role of auxin in case of endosperm, so this endosperm proliferation mainly requires auxin. So this auxin in case of endosperm are mainly paternal specific factors or PEG, paternally expressed genes. So from the isolation of the different auxin mutants, it is known that it is the paternal expressed genes which are responsible for synthesis of auxin, which will help in endosperm proliferation. Proliferation. Okay. So this is from the mutants that have been isolated from the auxins from the experiment that has been conducted in those mutants. It is observed that out of maternal and paternal expressed genes, it is the paternal expressed genes which will contribute for the endosperm proliferation. So rest of the things you need not to worry about. Okay, just make sure to remember the function and how it is synthesized and when it is synthesized. These things will be more than enough. So I have just listed out the things. So if you want it, you can just read, or whatever I have said or whatever I have explained, it is more than enough. Okay. So mutants, few mutants I have mentioned it here. You can go through the mutants and you can know like how they are determined, that whether it is before germination or before fertilization or after fertilization. This is about auxin. Have you heard about Avena coleoptile test? This is something which is related to auxin. Has anyone read about this Avena coleoptile test or oat coleoptile? So what is this Avena coleoptile test? If you remember, it was given by F.W. Went, or sorry, F. Went. Yes, it is in oat. It's a oat coleoptile or Avena coleoptile test. It is for auxin. It is something related to auxin. And what is it? Yes, it is F. Went. Yes, very good. So this coleoptile test by F. Went, he gave the auxin distribution. How the auxin moves in case of cells. So this is what he has given. The F. Went in 1926. He conducted the experiment in the oat coleoptile, where he came to know about how the auxin transport and distribution is taking place between the cells or cell membranes. So it is mainly by, yes, it is Havana curvature. Okay, phototrophic bending of coleoptile. Okay. So just remember the things which I will discuss now. You not to worry about much more in detail, because these are certain class of experiments. If you have to understand all these things, you have to go through the complete experiment, which is not at all required for you. Okay, fine. Very good that you at least know all these things. So this is mainly like auxin distribution or the transfer is mainly because of two methods. One is fast and one is slow. Fast method is a non-polar transport through the phloem, and slow method is cell-to-cell polar movement. And this auxin distribution or the transportation is mainly because of two main important things. One is pin proteins, which I mentioned it here, and secondly, it is mainly because of protonation and deprotonation. How the auxin is being transported or distributed among the cells. Okay. So why the name pin proteins? The mutants of these proteins will form a pin-like structure. That is the name. That is why the name the pin proteins. So these pin proteins and protonation and deprotonation will have the important effect or important part to play, or important role to play in case of auxin distribution. You need not to worry about all this experiment. Okay, I won't be explaining this all. It is for MSc, not for you. Yeah. Next, we have talked about endosperm development. What about embryo development? So while discussing the embryogenesis, I told you about basal cell and apical cell, right? So who will decide like which cell will be basal and which will be the apical cell? And who will decide its apical cell will form the embryo proper and basal cell will form the suspensor? So this is all because of the auxin. So this auxin controls the embryo polarity and the patterning. So as I told you in the previous class, there are different patterns of that you see in the embryogenesis. That is based on the divisions, whether that division is vertical, horizontal, or transverse, right? So how it is happening, there will be different patterns. So this patterning and the polarity is mainly controlled by the auxins. Anything left? Okay. So basal cells, what they will form? They will form the suspensor cells, and apical cell will form the embryo proper. Okay. So this is all about the auxin, which I have discussed here. So just go through the text that I have been written here. So if you don't understand anything, you can just contact me later. Okay. So, but just to keep it simple and basic, I just explained only the. I given the importance only to the auxins, their origin, their synthesis, and important function. Okay. So next is abscisic acid. So when this abscisic acid will be synthesized in the case of seed development, so the ABA content will be low in the early development, but in the mid development, in order to avoid the germination, again the concentration rises, and at last, once the seed filling is complete, and if it is completely viable after germination, if the condition of a seed is such that it will be completely viable after germination, so that is during the seed maturity, again the concentration of this ABA will decrease and the GA will increase. So this is how the germination occurs. So soon after the estro differentiation, the seed will have the ability to germinate. So minimum germination or the basic germination can occur soon after the estro differentiation. But if they germinate, they will be non-viable. Why they will be non-viable? Because they will be they are not ready to provide the nutritions. The tissues have not filled with the seed seed reserves. So that is why they are non-viable. So in order to encounter this, so the ABA content will be high at that time, which will inhibit the germination. Okay. So ABA content, the earlier in the earlier slides I told you about, the higher the ABA content, there will be low GA content. So this coinciding factor or this coinciding situation was proved by Illost in 1995. So the increase in the concentration of the ABA, which is coinciding with the lower concentration of the germination, was proved by him in the 1995. You can just observe this figure where you will be able to understand this better. So if this is the germination percentage, at the mid there will be increase in the germination ability, but due to the increase in the ABA content, this germination ability again decreases. As I told you, the ABA synthesis will occur from the mid stage of development or mid stage of seed development. Soon the ABA synthesis has started, there will be a decline in the germination ability. So this is mainly seen in case of abscisic acid. So I told you about the maternal ABA and the zygotic ABA. So why the maternal ABA is required? So this is to stop the precocious germination. Secondly, why the zygotic ABA is required? So this ABA is required to stop the precocious germination and then the dormancy induction. So these are the two different that this ABA should play a role in. So it has been observed that there are total of two peaks that have been observed in case of seed development. Two peaks of what? Two peaks of ABA that are being observed in case of seed development. So these peaks are generally in the 10 days after flowering and 16 days after flowering. So ABA synthesis in the zygotic and maternal tissues is the first peak at 10 days after flowering, and the second peak is the zygotic tissues after 16 days after flowering. And I told you the reasons why this maternal ABA is required and why the zygotic ABA is induced or synthesized. And the last chart says about the coinciding of germination or GA content and the ABA content, where you can exactly see here wherever the GA content is high, not directly GA, they are not mentioned about GA, but they are mentioned about germination percentage. If the germination percentage is high, it simply means that GA content is higher, because gibberellic acid is very necessary for germination to take place. So whenever there is a higher germination percentage, at that point there will be low ABA content in the cell or in the seed. So wherever the ABA content is high, again there will be a low GA content. So which was by Hilhorst in 1995. Okay, so this is all about the ABA content. So this how they are all been discovered. This is by just by the study of the different mutants that were available for ABA inducing genes and auxin inducing genes. Clear? Any doubts in this plant growth hormones? Yes. If you have any doubts, you can ask me now, or you can just continue with the next part. Okay. So till now we have discussed about the different plant growth regulators that are affecting the seed development. So now the complete development is over. So we have started our discussion from the gameto or sporogenesis till the seed development and different stages that are involved and different factors that are influencing them. So now the complete seed is formed. So why this seed is essential? So this seed is essential for four main reasons. So first one is establishment into the plants, food for human and animals, of course, and for the genetic conservation and for the efficient and profitable agriculture. So that is why we give the prominent importance to these seeds. So if I talk about the history of seed plants, so seed plants begin around 360 million years ago, and there are such some errors like eras. If you just see the historical timeline, so these particular seed plants have started in Devonian era, and they have exploded in Cenozoic era, and they are thought or they are thought to be emerged from progymnosperms. So the present day angiosperms and gymnosperms are thought to be emerged from the progymnosperms, which are nothing but the spores. These are the simple spores from which present day angiosperms and gymnosperms have been evolved. So if I talk about the classification of the seed plants, it is from the division Spermatophyta. So, able to write it very clearly. Okay, I'll just show it. So it is from the division Spermatophyta, which are nothing but the seed-bearing plants. So this particular division is subdivided into angiosperms and gymnosperms. These are naked seeds, and these are true seed-bearing plants. So again, these gymnosperms are classified as a monocots based on the cotyledon, which are single cotyledon and two cotyledons. So there are several differences between the monocots and dicots which you need to take care of. Okay. So I've listed all the difference. Just go through the difference. I know like you will be studying all these things from class 11th and 12th, but make sure to remember it now. So I've just also mentioned all the difference between the monocots and dicots. I may have missed some few, but all the major or important differences I have mentioned it here. So the seed is nothing but the fertilized matured ovule, which is made up of embryonic plant, stored food, and productive seed coat. What are the important parts of the seed? It's embryo, endosperm, and the seed coat. So can anyone between the angiosperms and gymnosperms? First one can be in case of double fertilization that we see in specifically in angiosperms. What are the other differences? So these angiosperms have the complex reproductive structure. So this is the other difference. And in case of angiosperms, you see the carpels are usually fused at the margins. When you just see the floral structure, you get to know about it. Next, the receptacle, which will be below the ovary, it will encircle this particular ovule. The ovules will be encircled or enclosed by the receptacle. So these are the major differences between gymno and angiosperms. So now we can talk of two different types of seeds, endospermic and non-endospermic. I think I have discussed it in the previous class also. The ploidy of endosperm. Yes, ploidy of endosperm, it is 3N. It is 2N in case of, it is N in case of gymnosperms. Yes, very good. So I think I have talked about the endosperms and non-endospermic seeds, right? So where the in case of endospermic seeds, we see that endosperm is persistent, it is not consumed, and it acts as a storage tissue. So this is majorly observed in case of monocots. So in dicots, we see that endosperms is utilized during the development, and the cotyledon is the storage organs. So though I have mentioned only two examples here, I have the complete list. The next page. So make sure to read it and remember it, because you might expect some direct questions in case of MISO following, or simply in case of MCQs, where they will be asking which one of the following is a non-embryon, like endospermic seed or non-endospermic seeds, all these kind of questions. So make sure to remember it. So endospermic monocots are includes different families, which are Poaceae, Orchidaceae, Singulaceae, Musaceae, and Ericaceae. So also you might get some confusion here, where within the orchids and within the palms, you will also get some varieties which will be non-endospermic monocots. So wherever there is a confusion, they won't be asking such type of examples. Only wherever there is a clear-cut information or clear-cut difference that this is endospermic and this is non-endospermic, they will ask those type of examples. So make sure to remember those. So this is generally in the grass family, where these are the cereals. All cereals are endospermic monocots. You remember all these things. And we have certain non-endospermic monocots. Though generally all the monocots are endospermic, we have certain non-endospermic. So I listed them here. Just go through that. So again, the dicot. When we take off dicot, there are rare or there are few dicots which are again endospermic. So this is the list. The rest of all will be, or whatever dicots are there, they will be non-endosperms. So these are just examples. It includes all other crops, which I have not mentioned it in these three tables. All other crops will include in this fourth table, if it is a dicot. Okay. Anything left? Seed coat. I think I also talked about the seed coat. Seed coat is mainly made up of testa and tegma. Testa is inner part, outer part, and tegma is inner part. So we have integments which will arise from the chalazal end, and these integments are again divided into bitegmic, unitegmic, and ategmic based on the presence of the integments. If there are two integments, it is bitegmic. If it is one, unitegmic, and if it is there are no integments, then it is called ategmic. Okay. So the functions of seed coats is very important for you. Just go through the functions once. Okay. Let us talk of non-embryonic storage tissue. So perisperm is a one such non-embryonic storage tissue which will act as storage in case of monocots. So this is generally maternal in origin, and this is synthesized by the nucellus itself, and the ploidy is 2n, and it is generally short-lived. And when I talk about the embryo has embryonic axis, plumule, radical. So if I just talk of monocot and dicot embryo, you just see the difference here. So it consists of the embryonal axis, plumule, apicotyl, radicals, testa, and storage cotyledons. This is again same here, but only thing is cotyledons, how they are dispersed. So you see the big endosperm in case of monocots, and the cotyledon is nothing but a scutellum in case of grass family. Single cotyledon is nothing but a scutellum in case of grass family, and the seed coat, which is nothing but a pericarp. These are the differences between the embryo of monocot and dicot. Yeah. So this is all about today's class. In the tomorrow's class, we will discuss about storage reserves in the seed. So we'll talk of synthesis, and we'll talk of mobilization. Synthesis, mobilization of storage reserves, and lastly we'll talk of dormancy. Different types of dormancy, what they mean, and what are the different roles, and which are the methods to break this dormancy. All will be discussed under the dormancy topic. So we have only left out with two different topics that is need to be discussed in case of seed physiology. Okay, we'll do it in the next class. Because if I start seed reserves in the seed storage reserves in the seed, or still seed storage reserves, I won't be able to complete the synthesis and mobilization at once. So it will be again some gap in between. I don't want that gap, so we will do it in the next class. Any doubts from today's class? Okay, if there are no doubts, you can meet in the next class. Thanks for attending. And all the notes will be available for all the master course students and students who are taken this seed science and technology course. So make sure to download it and read it. Okay. So if you get any doubts in case of notes or in case of live classes or recordings, you can just contact me on the WhatsApp anytime. So thanks for attending. We'll meet in the next class.